My name is Federica Ungaro. I'm the head of the Experimental Gastroenterology Laboratory at San Raffaele Hospital in Milan. As a laboratory, we have an interest in investigating the pathogenesis of inflammatory bowel disease, which is a class of chronic inflammatory disorders, mainly including ulcerative colitis and Crohn's disease. On behalf of our authors, today I will show you the data from a recent publication on the Gut Journal investigating the role of the gut virome-derived factor, HBX, during ulcerative colitis pathogenesis. The gut virome is a component of the gut microbiota made of both bacteriophages and eukaryotic targeting viruses. Gut virome dysbiosis has been already demonstrated to be involved in inflammatory bowel disease pathogenesis, but the mechanisms beneath are still unknown. Bacteriophages, for their nature, are able to interact directly with bacterial cells, thus shaping their compositions, while eukaryotic targeting viruses, for their nature to infect mammalian cells, including those from the host, are able to shape directly, most probably, the human cell functions. So, the aim of our work is to address specifically what is the function of eukaryotic targeting viruses in contributing to the ulcerative colitis pathogenesis. In this work, we identified the orthopadnavirus-derived factor, namely HBX, as associated with UC pathogenesis and defining a specific cohort of UC patients. For this reason, we isolated both RNA and DNA from UC patients-derived endoscopic biopsies. We detected HBX encoding transcript in about 50% of UC patients analyzed, while at the level of the DNA, only 4% of patients resulted as positive. These results are in line with the life cycle of orthopadnaviruses, which exist and replicate as episomal DNA and only rarely integrate into the host genome. We also verified whether HBX positivity in the colon was associated with the clinical characteristics of these patients, but we did not find any correlations, neither with the anti-HBV serum positivity. Of note, by overexpressing HBX in mouse colons, animals developed colitis-like symptoms, showing colonic ulcerations after 20 days of treatment. This proved for the first time the concept that a virome-derived factor may be an inducer of intestinal inflammation. Furthermore, going deeper into the cellular mechanisms, we induced the HBX overexpression in epithelial cells in vitro. We observed that HBX overexpression in epithelial cells increased the proliferation rate of these cells and increased also the epithelial barrier permeability compared to the control cells. This was also confirmed in HBX overexpressing organoids that showed upregulation of stemness markers and downregulation of epithelial barrier molecules. These results are in line with ulceration that we already observed during experiments in vivo. Furthermore, at the mucosal level, the induction of the HBX expression in vivo alters the immunomedia compositions by reducing the number of CD8 T cells, dendritic cells, and neutrophils, suggesting that HBX reduces the physiological response, ultimately favoring mucosal colonization by the other commensals. Finally, we asked whether HBX inhibition in vivo may serve as a therapeutic option. So, HBX overexpressing mice were treated with HBX targeting siRNAs, which reduced the colitis-like symptoms in vivo. In conclusion, our results showed that HBX impairs gut mucosal functions by altering the epithelial cell-forming barrier, that favors in turn the translocation of the other commensals and reduces the host response to pathogens. And this can happen in a defined cohort of UC patients positive for HBX. Future therapies targeting specifically HBX may be useful for treating inflammation in a selected cohort of UC patients. This work was possible thanks to the collaboration of all authors, the support from our institutions, and also from the different not-for-profit organizations that funded our study.